This is the lecture for Lab 4 on organic molecules. Organic molecules are the group of molecules that make up all living things. And organic molecules are based on hydrocarbon skeletons. And the hydrocarbon refers to the atoms that compose the skeleton, the basic framework of the molecules. And that skeleton is mainly composed of carbon and hydrogen. Here are some examples of different types of hydrocarbon skeletons that make up the basis of organic molecules. The hydrocarbon skeletons can vary in length. They can be very, very short, or they can be very long. The skeletons may have double bonds or single bonds, which can make different characteristics for the molecules. Also, they may be branched or unbranched. And lastly, they can be arranged in rings. So with these hydrocarbon skeletons, you get a lot of variety in the basic structure of the molecules, which can help give rise to the variety of organic molecules that make up living things. In addition to the hydrocarbon skeletons, which make up the basic structure of the organic molecules, there are also functional groups. And functional groups are groups of different types of atoms that give the molecules different characteristics. And there are many different types of functional groups found on different types of organic molecules. And these are just a few examples. The hydroxyl group is an OH group that's found mainly in sugars. The carbonyl group is a carbon double bonded to an oxygen, and this is also found in sugars. The amino group is composed of nitrogen and two hydrogens, and this is found mainly in amino acids, which form the basis for proteins. And lastly, the carboxyl group. This is a carbon bound to two oxygens and a hydrogen, and this is found in amino acids and some fatty acids. Dehydration synthesis is the process by which organic molecules are formed. Organic molecules are very large molecules that are formed by linking monomers together. A monomer is a single subunit of a particular type of molecule. Polymers are chains of monomers that have been linked together. So in general, to make an organic molecule, monomers, similar monomers are joined together to form polymers. And this is an example of a dehydration synthesis reaction. Here you have a short polymer, and you are going to add this monomer, a similar subunit, to that larger structure. In order to do this, a covalent bond bound to a hydroxyl is broken. This bond will be broken here. And a bond on the monomer to a hydrogen will be broken here. And that will allow these two subunits to now form a covalent bond to each other. Then that hydroxyl and that hydrogen will bind together to form a molecule of water. So this is why this process is called dehydration synthesis, because during this, in forming a larger molecule, you are going to remove a molecule of water. The opposite of dehydration synthesis is hydrolysis. And hydrolysis is the process where polymers are broken down into monomers. So when you take a larger molecule and you are breaking off those subunits. This is an example of a hydrolytic reaction. So here's your larger molecule, and the process is going to involve breaking off this last subunit. In order to do that, you need to break this covalent bond. So you break the covalent bond, and then you will need atoms to fill up that space. So the atoms that are available will come from water. So a molecule of water is broken in half. The hydrogen will make the covalent bond to one subunit. The hydroxyl will make the covalent bond to the other subunit. So because during this process, it appears as if a molecule of water is being split, this process is called hydrolysis, so water splitting. Now I want to go through the 4 types of organic molecules that compose all living things. And these 4 types are carbohydrates, lipids, proteins, and nucleic acids. So, beginning with the carbohydrates. The main purpose of carbohydrates in living things is to provide energy and structure for the living thing. And you probably know that if you need a lot of energy for a sporting event, often you will carbo load a couple of days beforehand so you will have enough energy. So carbohydrates, this provides energy for the living organism. And it can also help provide structure, especially in plants. Monosaccharides are the basic subunit of the carbohydrates. Mono means one, saccharide means sugar. And an example of a monosaccharide is glucose. This is also known as blood sugar. So glucose is the type of sugar that provides energy for humans and other animals. A disaccharide involves 2 monosaccharides bound together, and the most common example is sucrose, and this is table sugar. Last type of carbohydrate are the polysaccharides. Poly means many, saccharide means sugar, and these are long chains of glucose molecules bound together. And a common example is starch, and you find this in a lot of carbohydrates, potatoes, pasta. They're very starchy foods because they contain the polysaccharide starch. Monosaccharides are the basic building blocks for carbohydrates, and this is an example of the structure of glucose. And as you notice, atoms that make up glucose or make up the carbohydrates are mainly carbon, hydrogen, and oxygen. And glucose tends to have this ring structure. Here are some examples of polysaccharides. And remember, poly means many, saccharide, sugar. So these are many of these glucose molecules bound together. And a couple examples of polysaccharides. Starch is found mainly in plants, and it's for energy storage. Glycogen is found in animals, often in muscle tissue and in the liver, and it's also for energy storage. And another example is cellulose. Cellulose is found in plants, and it provides the structure for the plants. The second type of organic molecule are lipids. And the main purpose for lipids is energy storage, long-term energy storage. Many lipids are composed of fatty acid chains bound to a glycerol. Triglycerides are where 3 fatty acid chains are bound to 1 glycerol. And the fatty acids can either be saturated or unsaturated in their structure. Phospholipids are an example of another type of lipid, and these are the lipids that are found mainly in the cell membrane. Steroids are also lipids, and steroids are molecular messengers that help coordinate the behavior of different tissues in the human body. This is an example of a triglyceride, which is composed of 1 glycerol and 3 fatty acids bound to that glycerol. These 2 fatty acids are saturated fatty acids because all the bonds between the carbons are single bonds. This is an example of an unsaturated fatty acid because there is a double bond between 2 of the carbons. The third type of organic molecule are the proteins, and the proteins have many different functions. One of the most common functions that we talk about in lab are enzymes. Enzymes catalyze biochemical reactions in living cells. Also, proteins serve to help form the structure of living things. They help facilitate movement and also regulation. Amino acids are the monomers that make up proteins. So these are the similar subunits that are linked together to form polypeptide chains. So the polypeptide chain is linking together of amino acids to form the protein. And the basic structure for the amino acid is that every amino acid has a central carbon. Then bound to it is an amino group. On the other side, a carboxyl group. Then there is a hydrogen, and the last group is a side group. And this is a functional group which gives every amino acid a different characteristic. Protein structure is very important to the function of a protein, and protein structure can be a little bit complicated with 4 different levels. The first level of protein structure is the primary structure, and this is the order of amino acids that are linked together in one chain. Secondary structure is how that chain of amino acids starts to hydrogen bond with other amino acids in the chain, and 2 different basic shapes can be formed, an alpha helix and also a beta pleated sheet. The third level of structure is called tertiary structure, and this is how that whole chain folds in on itself to form a 3-dimensional structure. And the last level of structure is called quaternary structure, and that's how that polypeptide binds together with other polypeptides to form a functional protein. The last type of organic molecule that we will discuss are the nucleic acids. And the nucleic acid's main function is to provide information for the cell. Nucleotides are the monomers that make up the structure of nucleic acids. So the nucleotides are linked together to form the DNA and RNA, which are examples of nucleic acids. And the DNA, of course, is the genetic material that directs the function of a cell. This is the basic structure of a nucleotide, and the nucleotide has a phosphate group, a sugar group, and a nitrogenous base. And DNA has 4 different options for nitrogenous bases: adenine, guanine, cytosine, and thymine. And one of the differences between DNA and RNA is that instead of thymine in DNA, RNA has uracil. DNA is the molecule of information for the cell. It's the genetic material. The genes on the DNA direct the function of the cell. In terms of structure, DNA is a double-stranded helix with complementary base pairing, where adenine always binds across from thymine and guanine always binds across from cytosine. So this is the structure, a double helix. So double helix, 2 strands of nucleotides. So this is an example of a strand of nucleotides where the nucleotides are bound together. Complementary base pairing, adenine always lines up across from thymine, and cytosine always lines up across from guanine. In lab, you will perform experiments to determine the presence of different types of organic molecules. And again, because these molecules are so small, we cannot see them with the naked eye, so we need to use indicators that will change color in the presence of the different types of organic molecules. Benedict's reagent is an indicator that will detect the presence of monosaccharides in a solution. Iodine will detect the presence of starch. Hydrophobic paper will be used to detect the presence of lipids, and the Biuret reagent will detect the presence of protein. When performing these experiments, it is very important to include controls. The first type of control that you need to include is a positive control. And a positive control is a sample where the substance is present. So if you are performing a starch test, your positive control will be a sample that absolutely contains starch and should react in a positive manner. The second type of control is a negative control, and the negative control is a sample where the substance is absent. And the typical negative control that is often used for most experiments is water. The Benedict's test is used to determine the presence of monosaccharides in a solution. And when you add the Benedict's reagent, it will change color depending on the amount of monosaccharides present. If the solution is blue, that means no monosaccharides are present. If it starts to turn green or yellow, there are very small traces of the monosaccharides. As it moves to orange, there are moderate amounts, and a dark red color means large amounts of the monosaccharides are present. Iodine is used to determine the presence of starch, and when starch is present, the iodine will turn black. So this is an example of a positive reaction. If starch is not present, the iodine will remain a brown color. So this is an example of a negative reaction. The presence of lipids is determined by using hydrophobic paper. A drop of the sample will be placed on the paper, and if the sample absorbs into the hydrophobic paper, then it is positive for lipids. If instead the sample beads up and is not absorbed by the paper, the sample is negative for lipids. The last test you'll be performing in lab is the Biuret test, and this is used to detect the presence of proteins. If the reagent turns purple, that is a positive reaction. If the reagent remains blue, that is a negative reaction.